Hello friends, today in this video we are going to discuss about the scientist Alexander Fleming, and here in this video we are going to discuss about his contributions to microbiology as well as in medical science. Okay, so here let's discuss about his contributions. Okay, so first thing, so these are his area of contributions. So that are the research on antiseptics, discovery of lysozyme and the discovery of penicillin, which is the most important contribution of Alexander Fleming. Okay, so we will discuss each of these topics one by one. Okay, so let's discuss about each of these topics. So first thing is the research on antiseptics. So he first established that antiseptic agents which are used to treat wounds and which are used to prevent infections were actually killing more soldiers and at the time of World War One than the infections were killing them. So more soldiers are killed by this use of these antiseptic agents than they are killing by the infections. Okay, so more soldiers are killed by these antiseptic agents than the infections were killing them. So what is the reason? So in that time the antiseptics which are used, that are the carbolic acid, boric acid, hydrogen peroxides, were failing to kill bacteria deep in wounds. Rather, they were in fact lowering the soldiers or patients' natural resistance to infections, because they were that antiseptic agents killing the white blood cells. So these antiseptic agents kill the white blood cells, which are the natural resistance power to the body to fight against the infections. So as these antiseptic agents kill those white blood cells, at the result that lowering the soldiers or patients natural resistance to infections. As a result, they kill those peoples or soldiers or patients by the use of these antiseptics. Okay, so Fleming demonstrated that antiseptic agents were only useful in treating superficial wounds or the surface wounds. They can be treated as antiseptic agents for the superficial wounds or surface wounds only, but were harmful when applied to deep wounds. Okay, so these are all his contribution or research on antiseptics. Okay, so the next thing is the discovery of lysozyme. This is an enzyme. So what is lysozyme enzyme? Lysozyme is an enzyme that lies the cell wall of many gram-positive bacteria. It is an enzyme that lies cell wall of many gram-positive bacteria. Okay, so Fleming had, so how he discovered this lysozyme enzyme? So when he treated a patients who were suffering from a head cold, Fleming had taken secretions from the inside of nose of that patient, and he cultured that secretion to see whether any bacteria are present on that secretion, or not. To find this, he cultured that secretion of the nose of the patient. Okay, and in the secretion he discovered a new bacterium which he called Micrococcus lysodeikticus, which is now known as Micrococcus luteus. Okay, so after finding this, one day he accidentally sneezed on one of the dishes of this bacteria, and a drop of mucus fell from his nose onto that bacteria, and he discovered that the mucus had dissolved the bacteria and killed it. So he tried the same experiment with the tears and got the same result. So Fleming at the end discovered the common factor of that mucus droplet was an enzyme which is responsible to kill those bacteria. So he named his newly discovered enzyme as lysozyme. Okay, so in this way he discovered the lysozyme enzyme, which is responsible to lyse the cell wall of many gram-positive bacteria. So in this way he discovered the lysozyme enzyme. So the next contribution is the discovery of penicillin, which is the most important contribution of Fleming. Okay, so by 1927 Fleming had been investigating the properties of Staphylococci. On 3rd September 1928, Fleming returned to his laboratory having spent on an holiday with his family. So before leaving on his holiday, he had stacked all his cultures of Staphylococci on a bench in a corner of his laboratory. Okay, so after returning from this holiday, Fleming noticed that one culture plate was contaminated with a fungus. One culture plate of Staphylococci is contaminated with the fungus, and the colonies of Staphylococci surrounding the fungus had been destroyed by this fungus, whereas other Staphylococci colonies which are farther from this fungus that are growing normally. So here we can see that this is the Staphylococci plate, and these colonies are the Staphylococci colonies. Okay, and this is the fungus. This is the fungal contamination. Okay, so here we can see that the Staphylococci colonies which are far from this fungus, they grow naturally, but the Staphylococci colonies which are near to this fungus, they are destroyed by this fungus. Okay, so after observing this, he got interest to study about this fungus. Okay, so he started investigating it, and he found it, and he saw that he was able to isolate the mold, that is the fungus, and he identified it as a member of the Penicillium genus, more specifically that is the Penicillium notatum. Okay, and he found it to be effective against all gram-positive pathogens which are responsible for diseases such as pneumonia, gonorrhea, meningitis, diphtheria, etc. Okay, and he determined that it was not the mold itself but some juice it had produced that had killed the bacteria. So the mold or fungus itself didn't kill the bacteria. The substance which are produced by this mold or fungus, that killed the bacteria, and he named that substance as a penicillin. Okay, so how penicillin rupture or lyse the or disrupt the cell wall of gram-positive bacteria, here we will see. So suppose these are the penicillin, and they bind with the cell wall of this gram-positive bacteria whose cell wall are thick. So thick walled gram-positive bacterial cell wall here, and penicillin bind to it, and this penicillin stop or inhibit the cell wall synthesis of this gram-positive bacteria, and as a result the cell wall burst. Okay, or rupture, cell rupture done. Okay, so in this way penicillin ruptured the cell of gram-positive bacteria. So this is his function, the function of penicillin. Okay, so the next point is the transformation of penicillin into medicine. So Fleming found it difficult to isolate the precious mold juice or penicillin in large quantities, but so he cannot make this penicillin as a medicine. He cannot produce or transform this penicillin into a medicine. So in early 1940s, Florey and Chain, two scientists at the University of Oxford transformed this penicillin into the medicine, and they were able to mass produce of this penicillin medicine for the use during World War II. Okay, and in 1945, Alexander Fleming shared the Nobel Prize with these two scientists that are the Florey and Chain. He shared this Nobel Prize in medicine or physiology with Florey and Chain. Okay, and the award was made for the discovery of penicillin and its causative effect in various infectious diseases. Okay, so in this way penicillin is transformed into the medicine by the help of those two scientists. Okay, so these are all the contribution of Alexander Fleming to microbiology as well as in medical science. Okay, thank you for watching this video.